received her bachelor's in psychobiology and BA in linguistics from the University of California, Los Angeles, followed by post-bac research at Yale University. She started her PhD training at Vanderbilt University, advised by Dr. Andre Bastos in 2021, and focusing on multi-area high-density laminar electrophysiology in non-human primates. Her projects are now investigating neurophysiological evidence of predictive coding and predictive routing along the cortical hierarchy, which she will talk to us about today, and predictive processing under loss of consciousness. And she'll be telling us about her work testing some implications of predictive coding theory by measuring electrophysiology in the brains of monkeys during an oddball task, where a few surprises are mixed randomly among a series of predictable stimuli. Sophie, please take it away. I've made you a co-host, so you should be able to share screen.
All right, thank you very much, Mark, for that introduction. Let me see. If I can manage to make this work.
Seems to be working.
And you want to switch the display setting into, yeah. Or...
You might want to share the other monitor.
Right. And
You're still on your.
How about this?
You're still on the. Okay, now you're.
It's good now. Yeah.
Okay. Perfect. Yeah. Well, thank you all very much for being here, and thank you for inviting me, Mark. I mean, it's really an honor to be here to be talking to you guys, and quite a heartwarming experience because I was lucky enough to have attended Mark's neurodata science course a few years ago, and quite a bit of this project I drew from the stuff that I learned from the course, so it wouldn't have been possible without Mark and his team's teachings. So it feels like a full circle to come back here and share with you all what I've done in my recent work. So this project investigates how propofol-mediated loss of consciousness disrupts predictive processing and how brain rhythms are involved in this process. When I talk to different groups of folks, people tend to be interested in one of three things: predictive processing, brain rhythms, and consciousness, which I know a lot of you guys care a lot about. So I'm going to touch upon all three of these elements today, but feel free to ask me questions about specifics of anything that I talk about, and I'll be more than happy to have a more in-depth discussion about any of it. Okay, so the whole idea of this project is based on the now quite commonly accepted idea that making predictions is one of the fundamental functions of the cortex, and I would argue in conscious processing. This can be very low-level predictions, like if you have the AC on in your room and it's one pitch for a while and all of a sudden it changes pitch or it becomes stronger, and then your brain notices it. While it was constant, it was as if your brain subtracted it away. You don't even notice it. And this could be something a lot more complicated, like making predictions about the stock market or making predictions about the weather. This is the picture that I got from this local Twitter account that are supported by volunteers who are meteorologists to help with severe weather in the Nashville region where I live at the moment. And it's funny because I gave this a similar version of this talk about a year ago, and this is from last year. Although now that I'm giving this talk to you folks, we actually just had a couple of tornadoes tour through this last weekend. So it looks like as long as I'm here, this screenshot will be relevant. So what these nice folks, life-saving folks, I would argue, have been doing for the community is basically making predictions, right? They look at these radars and models and predictions, and they make their own predictions of where the tornado and when it's going to hit, and what should people do, right? It's pretty amazing stuff what the brain does in terms of predictions. So how does this happen, right? It's hard these days to talk about predictive processing in the cortex without talking about predictive coding, which is a signal processing idea initially, which is called linear predictive coding, which basically the idea behind which is using already existing time series signal to predict and reconstruct lost signals or predict the next signals. Let's say in music production, or sort of you have an old record that you're trying to rescue, right? And in the 1990s, some people took this idea from signal processing and tried to implement this in a circuit that presumably can also be implemented in the brain. And this is the sort of Rowan Ballard 1999 flavor, and then later Spratling flavored idea of predictive coding. Namely, when an input comes into the brain, error signals are computed and then fed forward, and then predictions are computed upstairs and then fed backwards. Specifically in the format of inhibition. And because we're neuroscientists, we also would like to know how this is biologically implemented in the brain. So here's the toy model diagram to summarize how predictive coding could be implemented at the circuit level when you look at cortical sort of canonical microcircuits in the cortical laminar columns. So here I'm showing different layers of the cortex and different areas on the cortical hierarchy. And so at the circuit level, how predictive processing is carried by these cells and synaptic connections that represent basically these computing units. And people who study brain rhythms, myself included, also sometimes find that individual neurons are not the full picture. Sometimes rhythms or brain oscillations actually give us a better functional idea of what is happening in the brain. And this sort of leads to my advisor's work when he was a postdoc in Boston. Which essentially posits that lower order cortex sends feed-forward prediction information through both spiking and also gamma oscillation in superficial layers of the cortex. And then prediction is again computed upstairs and fed backwards in deep layers of the cortex in alpha-beta rhythm. And this is what we call the predictive routing model. And of course, this is all dependent on the idea that there exists, first of all, a cortical hierarchy, which I'm sure is not a new idea to many of us. And this obviously has been extensively researched by scientists like Feldman and Van Essen, and then later Markov using techniques like anterior and retrograde tracers to look at the actual synaptic connections between different brain areas that established a cortical hierarchy as shown here. And also there exists a functional hierarchy, and that different frequency bands, again, this is going back to the predictive routing idea, different frequency bands are dominated in bottom-up versus top-down information flow. So in the case here, I'm showing a graph, information flow operationalized as grandeur causality influence. So we can see that for bottom-up information flow, gamma band grandeur causality is much stronger. And for top-down, grandeur causality is stronger in alpha beta. Okay, so then we can tie this idea to this idea that the brain processes prediction and prediction error to everything that we talked about, like cortical hierarchy, functional hierarchy, and the involvement of rhythms, right? So my advisor, again, Andre Bastos, in 2020 published this idea that if you look at in a delayed match to sample task, if you have blocks of predictable versus blocks of unpredictable stimuli, you tend to see an increase in gamma power and theta power and a decrease in alpha beta power if you compare the unpredictable over predictable stimuli, meaning that prediction error is often associated with gamma power increase and alpha beta decrease, meaning that prediction is associated with alpha beta increase and gamma decrease. Okay. So again, this brings back to the idea of predictive routing, which posits that prediction error, now that you can map it to the specific brain rhythms, is sent through gamma rhythm, and then prediction is sent back through alpha beta rhythm in different layers of the cortex, right? So one of the things that we did for my recent paper is to challenge the system by using propofol, which induces loss of consciousness, or I should say mediated by loss of consciousness, as instructed by the anesthesiologist on our paper. And consciousness is something that we think of as being very important to the capability of the brain of making predictions and to brain functions generally. So propofol, as many of you guys will know, is a GABA A agonist, and it's widely used in surgical contexts to induce general anesthesia. So what we were interested in looking at, which I should say this experiment was done by my advisor Andre and his colleagues at MIT, and I was the data analyst on this project. So the point of this project, specifically in terms of analysis and experimentally, was to look at what happens to the predictive routing system and to predictive coding in the brain if you challenge the brain with propofol, leading to loss of consciousness, right? So generally, what we know about propofol, which was shown in Andre's paper in 2021, is that, first of all, it, not surprisingly, overall depresses spiking in the brain, in all brain areas, right? But also specifically in frontal areas, it seems that frontal areas are more affected by propofol than lower order cortex. So then the question becomes, what happens when you introduce an agent like this to disrupt consciousness and particularly disrupt higher order cortex? What happens to this feedback loop in the brain that is implemented to make predictions possible, right? So this can be investigated with what's called a passive auditory local global oddball paradigm, which is quite a classic in the field of studying prediction and prediction error. And this is from Bekenstein's paper in 2009. And I was recently at a conference of mismatch negativity, which is sort of a classical EEG signal of prediction error. And I would say half of the people at the conference was showing some kind of variant of using this paradigm. So it's really a classic paradigm that's used in people who study prediction error or mismatch negativity or any kind of surprise signal, right? So for people who are not familiar with this paradigm, I'll quickly introduce it here. So basically, in my case, monkeys are shown a series of 5 musical tones. So it goes beep, 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 clustered in 5. And so every 5 musical tones is a trial, right? So there are 4 different block types. Within the 5 musical tones, if they're all the same pitch, this is considered what we call a local regular trial, meaning that in the local environment within the same trial, there's no change in stimulus, meaning that there is no oddball, there's no prediction error, right? And a local oddball trial, by that logic, would then be beep, 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 boop. So the last change in pitch here would then introduce a prediction error or presumably introduce a prediction error, and the stimulus itself being an oddball within that trial. So it's called a local oddball, right? But we can also investigate something more complicated, right? Sort of at the pattern level. If you were to introduce a block type where most of the trials, let's say 80% of the trials, are a local oddball where the last stimulus is different, but then 20% of the time, you would introduce a trial that's a local regular, which makes it at the global level, at the pattern level, rare, therefore an oddball, right? And then you can counterbalance this. And you have a 2x2 design of 4 block types. Okay. So the experimental setup looks like this. The monkeys were first awake. The oddball paradigm is presented. Then they undergo propofol infusion, which induces initial loss of consciousness. after which they enter a holding dose of about half of the starting dose. Then the paradigm is presented again while they're under anesthesia. And then they come back to consciousness after being taken off of propofol. Okay. So I would add that this level of propofol infusion is sufficient for general anesthesia. So the animals were no longer responding to air puffs or tail pinch, and their eyes were closed, their heart rate was down, their blood pressure was down. So it would fit sort of the criteria for general anesthesia. Okay, so today I'm going to present data from 2 macaque monkeys. One of them had these 64-channel Utah arrays implanted, and the other one had these 16-channel laminar U probes introduced acutely on each recording session. So we'll be talking about data from a lower order auditory area. Comparatively to FEF, it's actually mid-level for the auditory processing stream, which has a healthy amount of auditorily responsive channel. And then the frontal eye field, which surprisingly has more than what you would expect, number of auditorily responsive channels. And then I'll also talk a little bit about the prefrontal cortex later. Okay, so let's first of all look at just the main effect of loss of consciousness or the introduction of propofol on spiking. So here I'm showing in TPT and in FEF the average number of spikes per second under the awake state versus anesthesia. So right off the bat, not surprisingly, you see that anesthesia generally suppresses spiking in both TPT and in FEF, and you can sort of see it does so a bit more in FEF than in TPT, and it does so both in stimulus response and in just a generic baseline period. Then let's quickly look at the main effective loss of consciousness on spectral power. So here is decomposed sort of frequency data. Here I'm showing TPT in green here and FEF in purple. And the general trend looks quite similar between the 2 areas. And what's important to note is that in the alpha beta band and in gamma band, in both of these frequency bands, it seems that there is a general suppression of power, right? And you see it quite nicely for these 2 sort of, as people who study brain rhythms sort of predefined frequency bands, but you see it functionally here as well. And also, interestingly, we also see an increase in slow wave power. So basically, if you look at the raw data, what happens is that under normal awake state, your local field potential will look very active, and there are a lot of very fast frequency oscillations. And then once you enter anesthesia, your local field potential becomes very mild, and they would have these really high amplitude but very low frequency, as low as like half a hertz, of just waves of your brain not being able to process information. And you see the spiking. If you look at the spiking next to these slow oscillations, it almost looks like your brain is only given very short sort of windows of opportunity to be able to spike, which happens in a lot of different altered sort of states of consciousness, like sleeping, and it's shown quite pronouncedly in loss of consciousness here with propofol. Okay, so let's look at sort of the main effects of loss of consciousness on a oddball processing, so prediction error processing, when you look at the general multi-unit activity, so a sort of congregate of what spiking is looking like in the brain. So here I've plotted out the 4 sort of mutually exclusive conditions in different colors. So up here we have TPT under awake state and under anesthesia. Down here we have FEF under the awake state and under anesthesia. On the x-axis, obviously we have time. So the the paradigm is presented as such, it goes beep, 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 and then another beep or a boop is introduced, right? So what you can see right off the bat is, first of all, both brain areas will adapt to the first 4 stimuli very well. So the first 4 stimuli are always the same, right? So TPT goes quite down as you go on into the 4 stimuli. But then in FEF, the adaptation happens, or the learning, I would argue, happens quite faster. The first stimulus was introduced, it responds, and then by the second one, it already sort of knows that this is old news, right? So under normal awake state, what we would expect to see is that if the last stimulus is a different stimulus as the first 4, you would see a sort of a resurgence of spiking, which happens here. So both of the local oddball conditions you see coming right back up at the last stimulus. Same thing here in FEF. I would note that here there is no main effect of global oddball. So if you look at the beep, 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 beeps and the beep, 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 boops that are both rare in their block structure, you don't see really a difference between the beep, 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 beeps and the beep, 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 boops that are frequent in their block structure, which I will talk a little bit more in depth in a little bit. But the interesting thing happens under anesthesia. So if you look at this auditory area TPT, under anesthesia, it first of all still sort of responds to stimulus. However, it doesn't seem to come back down. It sort of loses its ability to adapt to or to even learn from the last stimulus, and you know, nevertheless the pattern, right? And when your last stimulus is a change, so these two are the local oddball conditions, so the beep, 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 boops, it's still able to be released from adaptation, right? So it's still, if you believe in brain coding, then it still it still codes this local prediction error. However, your frontal eye field in the context of prediction and prediction error is completely sort of eliminated, right? So you see a tiny bit of response here, and then it's no longer able to pick out, quote unquote, the last stimulus being a change. So it's no longer able to register even the local prediction error. So this sort of corroborates with the idea that the frontal areas are more affected by loss of consciousness than lower order cortex. And I will sort of talk more about this mechanistically, but it does also look like the response to local oddball in lower order sort of auditory area seems to be more smeared out in time, right? It almost looks like it peaks and it's not being controlled or fed back or inhibited by anything else. So it's sort of let loose in a way, as opposed to under the awake state, the peak happens and then it comes right back down, right? And this effect is, of course, by percentage a lot higher than this effect compared to the regular stimuli, the first 4 stimuli. Okay. So this is just another way of looking at the same data, but here I have sort of grouped the channels, the individual recording channels, based on the significance of their effects of global oddball and local oddball. So the global oddball effect is on the y-axis here, local is here. So basically what I'm showing here is that these blue channels, for example, are the channels in TPT that had a significant local effect. A local oddball effect, meaning that would significantly respond to beep, 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 boops over beep, 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 beeps, but they do not have a significant effect of global oddball. And these green ones right here, this very few green ones right here, would be the channels that are able to significantly differently respond to the sequences that are rare in their environment versus frequent in their environment, but not to a local oddball. And of course, these black ones are significant for both, and these grayed out ones are within sort of the cloud of insignificance, right? So these are channels that seem to not care about local effect nor global effect. And this is same thing for FEF. And FEF, not surprisingly, has a few more channels that would respond to global oddball. And when we look at the same channels under anesthesia, I'm showing in the arrows here, on average, where did these previously grouped by significance channels go under anesthesia, right? So if we look at these blue channels, what happened to these channels in terms of local effect and global effect on average under anesthesia, right? So very interestingly, in TPT, we see that a lot of these channels almost gain their effect in response to local oddball. So a lot of these channels that were previously not significant for anything would sort of escape this insignificance boundary. These channels that previously had a global effect would also gain local effect and then lose a lot of its global effect, right? As opposed to if you look at frontal eye field, most of these channels that are previously significant for either effect kind of end up back in the box of not significance for both local and global effect. Again, this goes with the idea that frontal areas are more severely impacted by loss of consciousness, or I should say propofol-mediated loss of consciousness, and lower order cortex seems to have been facilitated by the loss of consciousness in its ability to respond to a beep, 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 boop, a local change of stimulus.
Excuse me, Sophie, can I interrupt for a clarifying question for just a second? When you talk about the local effect, perhaps I missed it, but that is the average spike count per second on that channel, or is that some other measure of activity?
This is a multi-unit activity.
So yeah, not a particular neuron, but on that channel, the total number of spikes.
Yes.
Okay, thank you.
Yeah, please feel free to interrupt me as I'm going to kind of clarify anything. I think this helps me and it helps the group, so I really appreciate that. And I guess I have not been checking the Q&A, but I'll be happy to sort of get to all of it at the end. Okay.
I'll alert you if anything stays in the Q&A for very long.
Okay, sounds good. Thank you very much. So I did mention that earlier, we did not see a main effect if you combine the global regular together and the global oddball together. There was not a significant main effect between these two sort of grouped conditions. However, many sort of more complicated population-based decoding methods have shown that previously before my project in the prefrontal cortex, you would be able to see decodability of existence of a global oddball in actually exactly the same in the same paradigm in macaque monkeys, but only in prefrontal cortex. I believe that paper only showed data from that one area. So I went ahead and tried the same thing. And this is very interesting. So this decoder, it's a multiple linear regression, is able to decode whether or not a local oddball has happened in areas TPT, in frontal eye field, and in prefrontal cortex. So in the blue here, I'm showing sort of the decodability in time after the onset of the last stimulus is introduced. I should also say, so you can see stimulus is also a regressor in this model, and it actually does quite well being able to pick out whether or not it was a beep or a boop, but it's not surprising, so I'm not showing the results here. And this decodability sort of persists under anesthesia. Oh, I should say my labels here are switched. So blue is actually awake and the dotted red lines are anesthesia. In all three of these areas, you can still retain some decodability of whether or not it was a local oddball, sort of across time, right? However, if you look at the decodability of a global oddball, meaning if you ask the decoder whether or not this last stimulus that was presented was frequent, therefore expected by the environment that it's in, or rare, therefore unexpected by the environment that it's in, you only see sort of significant decodability in PFC albeit very slight, and in FEF, in the first sort of a 100 millisecond onset, and not in TPT, which tracks because sort of your frontal areas are considered to be keeping track of these more complicated patterns, more so than your auditory cortex. And more interestingly, this decodability completely becomes eliminated under anesthesia in both FEF and in PFC. So you can see sort of this red dotted line here and here just hover around chance, 50%. And for the people who care a lot about the local global oddball paradigm, people will also know there's this thing called the pure global oddball. The pure global oddball is the case where if you have an environment, a block structure where your global regular is the local oddball, meaning that most of what you hear in this block is beep, 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 boop, beep, 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 boop. And when you're introduced a beep, 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 it's considered a pure form of global oddball because within that trial, you have not experienced a change in stimulus, therefore no local oddball, but it should be surprising to the brain. So does your brain pick this out? And unfortunately, in this data set, it looks like there is no decodability in any of these brain areas for this pure form of global oddball, not in awake, not in anesthesia. However, I would say after this project, I've moved on to this experiment where we put monkeys in an environment where they hear the local regular over and the local oddball, beep, 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 boop. Actually, no, in this case, it's a visual experiment. So they see sort of gradings of different orientations, and they're presented with the local oddball over and over again for days on end. And then on recording day, we introduce 20% global oddball, which are local regulars. So 20% of the time they see all four the same visual stimuli. And there's some sort of evidence that they do encode this pure global oddball, but it emerges in higher order cortex. But I don't want to spoil too much. That work is in progress and it's actually on bioRxiv right now. Hopefully when that comes out, I can talk to you all about it again. Okay. So since I'm one of those people that care about brain rhythms, let's quickly talk about sort of the effect of different brain oscillations in the context of these oddballs. So here, because I didn't see a main effect of global oddball, I'm here just showing local oddball minus not local oddball, right? Local regular. So if you look at the awake data up here, in both TPT and in FEF, we see this sort of classic increase in gamma and decrease in alpha beta, which I sort of introduced alongside the predictive routing model. So the warm color here means that there had been more power for prediction error. And the cooler color here is there had been more power for prediction. And you see this very well in TPT and somewhat in FEF. So what happens under anesthesia? Under anesthesia, in TPT, it looks like there is still an increase in gamma, but the decrease in alpha beta goes away. And this increase in gamma becomes stronger comparatively in percentage, and it becomes more smeared out in time, which is quite similar to what we had shown in the spiking. And if you look at FEF, it becomes basically flat. So the brain rhythms are no longer doing its job, right? Again, going with the idea that the frontal cortex is more severely impacted by loss of consciousness, and with the idea that if you consider your frontal cortex to be important in giving feedback in the form of prediction to your lower order cortex, under anesthesia, since your frontal cortex is knocked out, your lower order cortex will respond to stimulus, to prediction error, but it's no longer inhibited. Therefore, this response becomes stronger and longer in time. Okay, so how does this happen mechanistically? One of the idea is that your local field potential brain oscillations is doing something in coherence with your spiking activity, such that your brain is sort of allowed different windows of opportunity to spike, as I was speaking about earlier, about the slow waves, right? So here, what I'm showing is that if you pick out in this case, for example, the alpha beta rhythm in the frontal eye field, and if you look at the fit between the alpha beta rhythm, its phase and its amplitude, to the spiking data, how big is that amplitude? Meaning, how big of an effect does your alpha beta rhythm have on your spiking? Yeah, here I'm showing sort of interareal results, and I've also done this within area, which I'll show in a second. And what is the displacement of this fit? Meaning that what is the optimal sort of phase at which of your alpha beta that your other area is able to do its spiking? Okay, so across the board, if you look at the amplitude of these fits, across the board it goes down significantly, meaning that under anesthesia, your brain rhythms, including alpha, beta, and gamma, is much less able to modulate your spiking, right? And also, very interestingly, we see that under the awake state, so in these sort of blue histograms, there exists an optimal phase at which the other area is allowed to spike. And you see this sort of more prominently in gamma, but also you sort of see it in alpha beta as well, is that the specificity of the phase at which the other area is allowed to spike becomes flattened. Therefore, your brain oscillation is less able to basically speak to the other area and modulate when it's able to spike, right? So this happens, so this becomes flattened in gamma and quite shifted in alpha beta. And a very similar thing happens if you look at within the brain area, sort of the alpha beta in TPT and its effect on spiking in TPT. Again, the amplitude of these fits across the board goes down, and the displacement of these fits is significantly shifted, right? So that brings...
Sophie, before you go on, could I ask a small technical question on this?
Sure.
So you've talked about the phase difference in terms of sorry, you've talked about the spike versus alpha beta oscillation coherence in terms of a phase shift. And, you're showing what looks like for the normal, for the awake state, that you're looking at differences of maybe pi/6 or something. And if you're thinking that that this is, let's say, an oscillation of 12 hertz, that would suggest a time constant of about 30 milliseconds. Is that roughly right?
I think so, yeah, if you look at it, yeah.
So that would be sort of you would think of that as almost like the conduction time, or do you think of it that way, or?
Interesting. I mean, it's easier to conclude that for the within area results than the interareal results, right? So, and since the interareal results still hold here, I would caution to interpret it biophysically like that. So
Okay, maybe we can talk about that later then.
Yeah, that sounds good. Yeah, because I'm interested in how this happens mechanistically, but the results that I'm showing here are sort of observational, right?
Sure.
Well, I should say it mechanistically speaks to the effect of a loss of consciousness on everything I've shown before, but how this happens at the synaptic level, I would be very interested to know, although I would hesitate to argue that based on my data here. Yeah.
Okay, thanks.
So I actually am coming to a conclusion here. What I've shown, sort of bringing it back to the canonical microcircuit that we're talking about in terms of prediction and prediction processing. Under the awake state, you see that the oddball stimulus comes in. It becomes computed in even the lower order cortex, and the gamma rhythm goes up, it gets carried upstairs, and it gets computed upstairs, and the prediction is fed back in alpha beta. But in the case of a prediction error, alpha beta would go down. However, under anesthesia, we know that higher order cortex from different modalities of evidence is significantly impacted, more so than lower order cortex. So your higher order cortex is no longer able to generate this inhibitory alpha beta to then inhibit your lower order cortex activities, such that when an oddball comes in, it becomes a bit haywire and it sends much more prediction error signal in both spiking and in gamma upstairs. However, this computation does not happen at the level of at the pattern level when a more complicated prediction error needs to be computed. Okay, so I will go to my acknowledgement slides. Obviously, I would like to thank my advisor, Andre Bastos, and all the collaborators and senior PIs at MIT, and of course, the funding sources. Thank you very much.
Thank you, Sophie. Very nice. I, of course, have several more questions, but I see other people do as well. So, Sai, would you go ahead?
I'm still thinking of my questions. I was trying to clap and I accidentally hit the raise hand.
Thanks.
Okay. Does anyone else have a question? Anyone else
have-
from the audience as well?
I have a stupid question.
No, nothing is stupid. Please go ahead.
So I think it's just because I don't do monkey research. So the FEF is used to as a frontal region because they have a reflex associated with the auditory stimuli or something. So yeah, I wasn't sure why, because when human, like everything is blurry, right? Frontal, there are a lot going on the frontal frontal eye field.
Right, yeah. So the frontal eye field, of course, in the monkeys is classically associated with saccades and some sort of higher level decision making, right? So in this case, again, I was not at the experiments when it was done. Although I would say I also work with quite a few of macaque monkeys that have frontal eye field chambers. So it's just a hotspot in monkey research. And in this case, it was quite surprising, but also fortunate that we have many auditorily responsive channels, and also you're able to decode sort of global oddball, global level of prediction error in the area, showing that it's still at the level of pattern recognition, if I could say so. It's doing something regardless of its processing stream, whether it be visual or auditory. Right?
Okay. Is there a reflex? Is there a like if I hear something here, do I have a reflex of jumping my saccading to the
Interesting. So I have analyzed the eye tracking data for this data set. I don't believe I saw significant differences. Therefore, the effect in FEF would not be from saccading or the reflex.
Okay, interesting. Thank you.
Yeah, of course. And of course, under anesthesia, their eyes are closed, and any effect would not be sort of from saccades, right?
All right, any questions from our audience? No. Sai, do you want to go ahead and ask your question, or shall I?
Yeah, so I'm kind of new to this topic, so I may have a basic question. So,
Of course, no worries.
yeah, have you tried to correlate any of these findings with like doing some more like connectivity analysis?
Yeah, so it is quite natural to think about connectivity, right, in this kind of context. I would say, first of all, the spike field, I mean, I would hesitate to call it coherence because coherence has its specific meaning, but it sort of gets at that, right? That effect that I'm showing interareally somewhat speaks to the connectivity changes under anesthesia, and clearly it goes way down. So I would say it then generally shows that under anesthesia, your brain becomes a bit less organized in a way, right? Different areas are less able to modulate each other, if that answers your question. And if you have any sort of specific connectivity results in terms of anesthesia that you think is relevant, I would love to hear about them, and I can talk to other people about them too.
Sounds good. Yeah, I'm just learning, so that was my question.
Yeah, thank you.
Any other questions from the audience or from the panel? Yeah, Barry, and then I have a few. We're not hearing you, Barry.
Your Zoom is unmuted, but something else has to be muted.
Yeah. Tell you what, Barry, why don't you figure that out? I will ask.
Or you could post it on the chat also, Barry.
You can also do that, yeah. So I have a technical question and a general question, which and the technical question may not have any reasonable answer. So, so first, one of your slides, you showed the decrease in or changes in power spectrum in decibels, I think. And you showed, for most of the beta and gamma rhythms that you were seeing about a 4 or 5 or 6 decibel decrease in the power. Yes, that slide, thanks. I noticed you've got a region that shows almost no decrease at all. It looks to be about 30 hertz, if I'm not mistaken, if that's the log scale. And there's some modeling studies of gamma rhythm that suggest that something like 30 hertz is kind of the natural oscillation rhythm of the cortex if under strong input. If you throw a lot of strong input at it, then the excitatory and parvalbumin interneurons will sort of oscillate in a network at about 30 hertz. And I'm wondering if you think there's any significance to that particular, that band, that maybe it's not being used for communication or something, so that it's not affected much by anesthesia. I'm just, guessing in the dark here, but do you think there's anything significant about it or not?
Right, that's very interesting because I almost had the opposite interpretation of what I'm seeing here. First of all, I would say that the modeling studies that show this sort of 30 hertz resonance was certainly, I would consider that to be relevant to the work here. And if I were to interpret it, not based on my results, if I read those papers, I would interpret that as a prediction error signal, right? Like input is a lot of times prediction error, right? Because it's a change from baseline. So it would somewhat even contradict what I'm seeing here, right? So here, when I see this sort of coming up to almost to no change, I interpret this as this is a sort of gap in the frequency bands that the brain doesn't use. So earlier, what I was saying was that it's quite interestingly and functionally, we see that the gamma band, which here I'm for the most part defining, and I also believe it's consistent with the literature that it's 40 hertz and above, and alpha beta is 18 to 30, I think here I've shown boundaries, are indeed functionally here the two dips in frequency, right? So yeah, now that you've mentioned the 30 hertz thing, as an experimentalist, I would trust the data that it looks like what's functionally relevant is 40 hertz and above. Although what you're saying is it makes sense, right? Like it looks like the 30 hertz is sort of a backbone of what the brain is doing, and it doesn't care whether or not you're under anesthesia, right?
Yeah, well, perhaps almost a biophysical phenomenon rather than a meaningful, but we can, again, let's talk more about that this summer, because that's something that, would take a longer time than we have here. The other question I did want to ask, and this may be a bit broader interest to other people on the call, is can you mentioned that you think, or your advisor or other people think that the alpha beta rhythm coming from prefrontal into sensory areas sort of carries in some sense predictions. Can you elaborate, or does anyone have any more specific concrete scenario? Are we talking about, neurons that are specifically suppressing neurons that would normally be responsive to a stimulus? Are we talking, so what, and why is it carried on let's say 12 hertz, which is I guess about a typical prefrontal to sensory sort of return trip time in a primate, I think. Are we talking about recurrent sort of probes and responses, and then that adjusting the sensitivity of, I'm just sort of wildly guessing as to what might a plausible mechanism for this conveying of predictive information be. And if someone has a more compelling metaphor, I'd be glad to hear it. I just haven't heard it.
Right. So I think I would go back to, first of all, the classical predictive coding hypothesis, which is very neuron driven, right? So the initial idea is that these prediction neurons are specifically synapsing onto the columns that have stimulus preference, right? Whether it be visual gratings or it be a certain pitch in the auditory cortex. And like I was mentioning, a lot of the work that I've been doing recently specifically looks at that. So we use very high density laminar probes in multiple brain areas at the same time in macaque monkeys to look at to sort of fish out these specific neurons and how they're actually what they're doing in V1, let's say. And from what I've seen so far, which of course we're still in the process of interpreting this data, so this could change by the time we talk again in the summer, there's very limited evidence from what I've seen that these sort of top-down specific prediction neurons exist. And you could even make the argument, sort of like the argument against Jennifer Aniston neurons, right? You don't have enough neurons in your brain to do that very specifically for every single stimulus that you encounter in the world, in the environment. So then we move from there to the idea that rhythms are doing this, right? And in my mind, a lot of this idea comes from studies in attention and working memory, alpha beta being this sort of top-down feedback kind of control role. And eventually we interpreted that as sort of inhibitory control, right, in the case of working memory, attention, and also prediction. Now, the specificity of what these rhythms can do is an open question, right? And of course, the mechanism of it, which is something that me and my lab, we've been also thinking about ways to study. So we recently started doing optogenetics in monkeys, where we specifically stimulate inhibitory neurons to generate certain rhythms. And we observe both locally and in other cortical areas simultaneously what happens and how is it that these specific rhythms are inhibitory and how specific it can do so. And so again, it's an open question. I wish I could give a more satisfying answer. I mean, I do think that when you want to have a brain rhythm, you need sort of ensembles of neurons working together, making it an oscillation, right? And presumably these ensembles can be projecting to other brain areas specifically and inhibiting specific columns or streams, and therefore making it stimulus specific. Although these are sort of just my on-the-go hypothesis, and we'll have to do a lot more work to figure out how that is. Does that answer your question? I hope I'm just
well, I think it shows that it's not really answerable right now, that nobody really has an answer right now, which is fine. I would like to think in terms of maybe, the cells in some prefrontal area becoming more synchronized with certain specific subnetworks in the, some cells, a subnetwork of cells in the prefrontal becoming more locally synchronized with cells in the sensory area, and then, test, in some sense, probing and then getting a feedback, depending on what's coming into those specific sensory cells, And then, if there's a rapid, that maintains a sort of stable dynamical point, or maybe a limit cycle that then gets disrupted, and then causes disrupts the balance of excitation and inhibition in both areas, so that's my hypothesis. So of course,
that's very interesting, and I think the data that I'm sitting on analyzing right now is capable of answering that. So I'm going to write that down, and then I'll work on that. Hopefully by the
and
summer,
if you want to
I'll
talk
have
more, I mean,
answers.
this is just off the top of my head. Also, Mungsen knows a lot about these kinds of things. But if you want to talk more about it, I'd be happy to either now or in the summer class.
Sounds good. Yeah, thank you so much.
Can you hear me now?
We can hear you now, Barry, and then Mingsen.
So my question is more general about human consciousness. There's a lot of talk these days about the fact that people go around predicting everything and not really seeing what's there so much, only changing the prediction if there's something strange going on. And this is sort of like the opposite of being very mindful, right? This is just kind of just like automatic kind of thing, and only reacting if there's something unpredictable that happens. So I'm kind of wondering, for instance, if this anyone has done the oddball paradigm when someone is on a hallucinogenic, which is supposed to disinhibit predictability and stuff like that, as a comparison, perhaps. So that's we can go with that question.
Yeah, that's very interesting. I can try to send, I'll look around and try to send some references, because I've met a lot of folks who use the exact same paradigm, and folks who are interested in pharmacology, and this paradigm has been used in all kinds of sort of altered states of consciousness. So let me write that down. But I would say that the theory makes it sound like everything is subtracted away when it is predicted. But clearly, sort of our experience is not so much so, right? You notice it less, but you still, on some level, the stimulus stream still comes in. So maybe that's the thing of the rhythm being not specific enough, or there's something else going on, right? Like maybe your brain maintains some kind of a feedforward stream while being inhibited in certain other streams. So that's interesting to think about, right? Because the brain obviously is always more complicated than your theory. I would also say that we've been working on training monkeys to actively respond to whether or not they had seen a global oddball. So sort of boosting up the you could call it an intentional factor, or the behavioral relevance of this global oddball, to see what then the brain does, what then the prefrontal cortex does, and what then your lower cortices do. It's a long process of training monkeys to report on something as complicated as global oddball, which makes me sometimes wish that I was working with humans, which I did a few years ago, and I decided to go work with animals with these sort of more dense recording techniques. But then, pros and cons of each model.
Humans kind of like propofol, as far as I've heard. Monkeys probably enjoy it too.
Yes, and hallucinogenics.
I just thought I did think this predictive theory is being overblown and a little too extreme, in that we don't see anything for what it's worth. We just see if it fits our prediction. I just thought that was a little extreme.
Nah, of course, when you what do you call it, when you have a hammer, everything looks like a nail, right? And I have many hammers. So I am sort of in the world of predictive processing and predictive coding, predictive routing, right? So when I see mismatch negativity, when I see oddballs, sometimes when I see like operant conditioning, it all looks like predictions to me, right? But I could be overgeneralizing. I could be overpredicting.
Yeah, I hope you have some screwdrivers too in the toolbox.
Yep.
Okay.
That's the next
I'm just going to mention that we are past 1 o'clock. So I think we're past our regular time, but let's just make sure we thank Sophy for an engaging conversation. And I don't see any other questions from the audience, so I'll just turn it over to Mungsen.